Good afternoon. I'm Dr. Brian Shapiro, a scientific content specialist at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Prevent Analysis Variability by Using NGS-Authenticated Microbial Genomes. Shift from Consensus to Discernible, presented by Mr. Andrew Frank. Mr. Frank is a bioinformatician at ATCC. In this presentation, Mr. Frank will provide an overview on ATCC's Enhanced Authentication Initiative. He will then focus on many of the underlying problems in consensus-driven genome sequences in public databases. Finally, Mr. Frank will introduce ATCC's new robust NGS and genome assembly workflow, which has been used to enrich the characterization of the biological materials in our collection. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome Andrew Frank.
Thank you, Brian, and thank you all for joining me today for our webinar series. Like Brian said, my name is Andrew Frank, and I'm a bioinformatician here at ATCC working on our new Enhanced Authentication Initiative. Of course, many of you already know ATCC and our mission. We're a nonprofit organization founded in 1925 with headquarters in Manassas, Virginia, and an R&D and services center in Gaithersburg, Maryland. Importantly, we're the world's largest and most diverse biological materials and information resource for microbes, where we have over 18,000 bacterial strains, including over 500 certified reference strains, in addition to many other materials like viruses, cell lines, and fungi. Today, I'd like to focus on the work we're doing on our largest collection, our bacterial strains. In today's webinar, I'll share with you ATCC's new Enhanced Authentication Initiative, which is ATCC's ongoing task to improve characterization of our biological collections with whole genome sequencing. I'll also be sharing with you parts of the ATCC Genome Portal, where we're making ATCC reference quality genomes available for public download and for use in your lab's ongoing analyses. So, to state the obvious, science is changing today. In the past decade, we have seen tectonic shifts in research practices, data generation, and how we use that data to solve our real-world problems. Specifically, we're now experiencing two paradigm shifts in the sciences, both impacting biology and microbiology specifically. Right now, we face the reproducibility crisis and the dawn of the genomics revolution at the same time. And of course, we know these two paradigm shifts don't exist in a vacuum. Said another way, can we leverage the data and techniques used within genomics to help solve the reproducibility crisis? If you know ATCC's work, the follow-up question was pretty logical for us. How can we apply technologies and techniques from next-generation sequencing and genomics to further enhance our own authentication processes? And furthermore, what are our customers doing right now to leverage genomic data? Let's walk through a scenario first where a scientist wants to authenticate the identity of ATCC 12228, a strain of Staph epidermidis. First, ATCC uses state-of-the-art polyphasic authentication using both bacterial phenotype and genotype information. However, we know bacterial taxonomy is rapidly evolving towards genome-based identification, moving beyond just phenotypic and 16S-based identification. Here at ATCC, our mission is to keep our promise to scientists that they're getting what they expect when they order a particular item. But let's say our scientist must get the genome for their ATCC item for analysis in their lab. What existing routes do they have to leverage the genomic data at this moment? Well, the scientists could sequence it themselves, either on their own instruments or they could send out a particular item for sequencing. But we all know this can be an expensive process, especially when not done at scale for many, many different strains at the same time. Additionally, in order for this to be reproducible, it must be done as close to the receipt of the ATCC item as possible. We all know labs share material that has been passaged 5 times, 10 times, and so on. ATCC items independently passaged that many times are no longer reproducible. Alternatively, our scientists could quote-unquote crowdsource the genome from a public genomic database and get the genome from some other lab that shared their sequence genome on a public database. But of course, this suffers from the same reproducibility problems as sequencing it yourself. But this is even riskier because if you let another lab do the sequencing for you, and you often need bioinformatics expertise to determine the quality of the genome you've downloaded and to ascertain whether or not you've actually got a contaminated genome. So at ATCC, we knew we had 2 major areas to address. First, how do we bring authentication into the genomics era and keep our promise to customers that we fully and accurately describe the ATCC items we're sending them? And second, for the customers that need genomic data, how do we effectively provide them with this data in a method that removes the uncertainty of the public genome crowdsourcing approach? This was the mission of our Enhanced Authentication Initiative, where we both enrich the characterization of ATCC biological collections via NGS sequencing and whole genome assembly, while providing our customers with these specific whole genome sequences of authenticated materials that you need in order to generate credible, reproducible data. At the end of September, we launched the ATCC Genome Portal, a collection of microbial genomics reference data sequenced by ATCC at the source. And these genomes are available for public download. So here at genomes.atcc.org, you can download whole genome sequences and their corresponding annotations. You can search for nucleotide sequences and view our assembly metadata and quality metrics. I'd like to take a brief moment and show you some of these features. Genomes.atcc.org is accessible from all major modern internet browsers. You can find the ATCC Genome Portal again at genomes.atcc.org. When you navigate to the Genome Portal, the first thing you'll want to do is log in or create a new account. Note that this account will be unique to the ATCC Genome Portal. So when I go ahead and click the login button on the top right, you'll see the option to either register for a new account. But right now, I'll go ahead and log in with my demonstration credentials. Great. Now that I'm logged in, I can now access the entire database of ATCC bacterial whole genome sequences housed at genomes.atcc.org. And you have a few options here on the homepage of how you can explore genomes. If you want to see a table of all genomes that we have available, you can click on the Genomes button in the top bar. If you have a particular search criteria you'd like to filter our collection by, you can type your search query in this box right here. And finally, if you want to jump into something we've recently published, you can select one of the genomes we've highlighted in this section below. Let's go ahead and click on Genomes at the top to see a table of all the available genomes on the portal right now. Right now, we have over 280 complete circularized reference quality genomes for ATCC's most in-demand bacterial strains available. These include many standardized reference strains, type strains for popular bacterial species, strains that are part of our global priority superbug collection, and strains that are part of our microbiome reference standards. From this table, you can easily see the taxonomic designation of an ATCC strain, a link out to the ATCC product page, where you can obtain the material directly associated with this genome, and a link to download the genome data right away. Let's go ahead and download the assembly.fasta file for this first Acinetobacter baumannii. Go ahead and click on Assembly, parentheses FASTA, and you'll notice that in order to use this data, you'll need to agree that it is for non-commercial use only. If you're interested in using our reference quality genomes for commercial applications, you can reach out to ATCC support and we'll guide you from there. I'll go ahead and hit agree and I can download that genome. Great. Right now, I'd like to tell you a little bit more about the processes that went into the creation of the genomes that you've seen right here. So let's say you're deciding between crowdsourcing an ATCC genome from a public sequencing database or using a reference quality genome from genomes.atcc.org. What are the key differences between these different genomes? Well, here at ATCC, we think of our genomic data as being differentiated in two key ways from public databases. First and foremost, we provide researchers with data that is sequenced directly from ATCC authenticated source biological material. For those of you familiar with our depositing process, we go through a thorough process to confirm and record details about the organism coming into our collection, along with performing the multiphasic testing I've already mentioned. In essence, you know our material comes with authenticity and traceability, two key ingredients for reproducible research. Second, when we're ready to produce a genome for one of our bacterial strains, we generate this high quality and complete genome by following state-of-the-art, data-driven, heavily quality controlled bioinformatics best practices. I want to spend some time going through these 5 steps in ATCC's bioinformatics best practices as seen below. The first critical step for us is extracting DNA. Here we use optimized proprietary protocols to get the best quality input material on a per organism basis. In essence, we want to avoid the common pitfall seen in public genomic databases of garbage in, garbage out bioinformatic data and results. If your input material is contaminated, your output genome will be contaminated, and that's the exact scenario we're avoiding. So to avoid bad DNA going on into the top of our workflow, we make sure we collect sufficiently high concentrations of DNA from the bugs we grow. We make sure that DNA is pure via nanodrop 260-280 ratios. And we make sure we capture the largest genomic DNA fragments possible. I'd like to highlight this last parameter. For us, obtaining the largest possible DNA fragment size is incredibly important for our genome assembly step, as I'll show you in a moment. Note that for some of our ATCC items, we're now making available the same DNA extraction used to produce genomes in the Genome Portal available to the research community. These DNA extractions, or NGS-Ready DNA, are part of a new derivative product line called the DX products. These products contain at least 10 micrograms of DNA and have DNA fragment sizes greater than 20 kilobases, and 50 of these DX items are now available. You can check these out at atcc.org/NGS-Ready. Once we've performed our DNA extraction, we go on to sequence on 2 state-of-the-art platforms. First, we perform Illumina short read sequencing, and then we perform Oxford Nanopore long read sequencing. Importantly, at this step, we impose another set of strict quality control thresholds to save only the highest quality reads per instrument. For both sequencing platforms, we go through a process called read trimming. And this is where we impose cutoffs on the Q score, or the so-called quality score, of the individual base calls or reads. In this case, the base call is an A, C, G, or T in the read. For Illumina, we only keep high quality base calls made by the instrument. In short, we only keep bases that have a better than 99.9% chance of being correct. For Oxford Nanopore reads, we're actually much less concerned about per base quality and more concerned about the average quality across the entire read itself. So here we average together the quality scores from each base and keep the read if the average quality score is above 10. This simultaneously also allows us to select for the longest possible reads going into our assembly. During the process of developing our sequencing technique, we wanted to make sure that we were able to repeat our sequencing results, especially across variables that potential members of the research community might care about. One of the first experiments we performed was to measure the consistency of our sequencing against the best available public genome for a set of ATCC items, with the prediction that we'd be able to consistently find SNP and indel differences between the source ATCC item and the public genome across different experimental treatments of the input library. When we mapped our Illumina short reads back to the public genomes from various experimental treatments, we found that differences between our sequence and the public sequence were consistent across A, DNA storage conditions, B, NGS library prep method, and C, even extraction method. In short, our sequencing results showed the detected differences between our material and the public material were consistent. While we're on the topic of sequencing, let's talk about how a member of the research community could leverage the data on the ATCC Genome Portal to test the accuracy of their own sequencing. If you're in an assay development or clinical testing business, you need to demonstrate that your NGS testing methods are accurate. In other words, you need to measure your own sequencing base calls against a true value. Let's broadly sketch what an accuracy test might look like for next generation sequencing. First, we need to start with some standardized, clinically relevant material. These Salmonella, E. coli on staph strains are all standardized strains available through ATCC. Second, your lab should perform DNA extraction, and then you should go ahead and sequence the material multiple times to get a measure of the precision of the next generation sequencing platform. Third, we should map the resulting reads from each sequencing run to a ground truth genome. But wait, what ground truth genome are we going to map these sequences back to? In the case of the ATCC Genome Portal, you'll have your answer as soon as you order your ATCC product. Let's jump to ATCC's website for a moment to demonstrate this. Here on ATCC's website, let's go ahead and search for ATCC 6538, the Staph aureus strain that I showed on that last slide. Go ahead and search 6538. You'll see a number of ATCC products appear. When you search for this item, you'll see that many of the search results have the genomic sequence available icon on the product, which actually leads you right to the reference genome for a given product. So here we'll scroll down, we'll find the certified reference material strain that we're interested in, ATCC CRM 6538, and we actually go ahead and click on the genomic sequence button. I'll pull this up in a new tab. You can go right here, and you can see a page outlining the specific genomic features of this reference quality genome. Let's go back to our product search for a moment. What I want you to notice is that any product derived from this source organism, 6538, also has this icon. So even if you're only interested in purchasing genomic DNA for this product, like from 6538DX, which is our new next generation sequencing ready DNA, right here, you can still go directly to its genomic sequence. Back on the genome page for 6538, in this view, you can see details about the genome assembly and annotations, which I'll talk about in a moment, right here, along with details about the source organism here, and the sequencing data used to produce this genome right here. Importantly, again, from here, you can instantly download the genome assembly, then use your lab's bioinformatics pipeline to map reads and determine the accuracy of your NGS testing method. Let's jump back into ATCC bioinformatics best practices. The next step is using data from both Illumina and Oxford Nanopore platforms to achieve high-quality, complete genomes. Here we perform hybrid genome assembly, leveraging the strengths of both platforms while avoiding biases and weaknesses of them individually. Here in this diagram, we show an example Illumina-only bacterial genome assembly all the way on the left. As you can see, it's quite fragmented, and the relationships between these fragments, or contigs, are unresolved, resulting in a tangled-looking assembly diagram. This is where our Oxford Nanopore long reads come in, which are game changers even though they're slightly more error-prone than Illumina reads. Here, Oxford Nanopore reads are first mapped to an individual tangled region within the Illumina assembly on the left. From this mapping, a path is traced through the short read contigs according to this long read alignment, a process called pathfinding. Once the optimal path through these pieces is found, the pieces are combined into what's called a bridge. As you apply long reads to tangled regions within the short read assembly and generate these bridges, you gradually resolve relationships between the short read diagram, eventually resulting in a circularized genome like the one you see above on the right. So how does this technique compare to other ATCC strain genomes available on public platforms? In this diagram, all ATCC genome assemblies are connected to their corresponding best public alternative assembly. In each graph, the ATCC genome is in purple and the public genome is in yellow. Looking at contig count on the left, we can see a downward trend in contig count, indicating more complete genomes than their best public alternative. Looking at N50 on the right, we see the correlated upward trend, indicating again that our genome assemblies are far more complete than their best public alternative. Note that for a number of items, the best public genome is roughly as complete as our ATCC reference genome. And so here at our project, we asked ourselves if there were other differences between our reference grade genomes and the public genomes not otherwise captured in metrics of genome completeness like contig count and N50. Let's consider ATCC 12228 again. Here we compared the ATCC genome assembly to one of the best genome assemblies publicly available for this strain. Here the public genome is also advertised as being a complete genome sequence. First, we asked if there were differences in chromosome structure between the two assemblies. Most definitely in this case. These areas in blue in the Mummer plot on the left indicate areas of the public assembly that have been incorrectly reversed as compared to our genome assembly, as confirmed by mapping our ultra-long Oxford Nanopore reads back onto our own assembly. Second, let's look at plasmid contents. We blasted the plasmids from the public assembly to our assembly's plasmids and looked for matching regions. If we had the same plasmid sets, all the plasmid graphs should be blue. Obviously, they're not. The public assembly actually has a completely different set of plasmids than ours. Again, highlighting the danger of assuming publicly available genomes are good proxies for ATCC material and highlighting the danger of sequencing ATCC materials away from the source. In the next step in our bioinformatics best practices, we wanted to make genome annotations available for users that were specifically interested in doing immediate gene level analyses. Of course, feel free to download one of our assemblies and annotate it using your own pipeline of choice, but we wanted to give portal users an opportunity to dive right into the annotated genes if they wanted to. Let's take a look at one example where we can immediately leverage the power of our genome annotations. ATCC hosts 57 strains of WHO priority pathogens, the so-called Global Priority Superbug Collection. These genomes are being made available on the ATCC Genome Portal, and these strains come with rich source metadata and susceptibility data, including MIC values and susceptibility profiles for targeted drugs. You can find more information about the Global Priority Superbug Collection at atcc.org/global priority superbugs. One superbug genome hosted on the ATCC Genome Portal is ATCC BAA 2803, Acinetobacter baumannii. Interestingly, in Acinetobacter baumannii, mutations of a specific gene called GYRA can confer high levels of resistance to fluoroquinolone antibiotics, such as Cipro. We can leverage the gene annotations on the ATCC Genome Portal to perform a search without needing to rely on slower sequencing search methods in order to find differences in the GYRA gene. Specifically, the ATCC Genome Portal can rapidly search for sequences of interest within our genome collection up to 4,000 times faster than an alternative search program like BLAST. Let's take a moment and see if we can find GYR in BAA 2803 and then find some mutational differences in GYRA among our Acinetobacter baumannii collection on the Genome Portal. Back in the portal, let's start by searching for BAA 2803. Of course, you can access any of our genomes by simply entering the ATCC catalog number into the search bar right here. Okay, that was easy. We found it and we can jump to this genome's individual page by clicking the View button right here. On the organism summary page, you'll see the susceptibility profiles that our global priority superbug genomes come with. And you'll notice that BAA 2803 is only susceptible to Cipro at concentrations greater than 4 micrograms per milliliter. Next, let's take a look at the Genome Browser tab, where we'll find a linear view of the genome assembly along with gene annotations in the table below. If you're interested in getting all the annotations in this table, simply click the Download Table CSV button, and you can get this whole summary in a format that you can easily open in Excel. Let me show that to you now. Here we go. Click here. This CSV file will open in Excel and any other program compatible with CSV files. And you can see that it comes with strain information, contig information, and importantly, amino acid sequence and nucleotide sequence. Let's go back to the Genome Browser page. You can see that for each gene in this table, we list the gene name, the gene's protein product, its position in the genome, and if available, the protein's enzyme commission number and the UniProt ID. We'll go ahead and search for GYRA, and we'll see if we get hits. Okay, here we go. We have one hit for GYRA, and the other is for GYRB, another related DNA gyrase gene. If we click this arrow on the right, we can jump to this gene in the linear view of the genome above, and we can see which genes flank GYRA. Finally, we can again download the table to get the sequence of this gene, or we can click and copy the nucleotide sequence here, like this right here, and then you can go ahead and paste it into the location of your choice. Okay, what if we started with a GYRA sequence we knew that was fully susceptible to Cipro and wanted to compare it to the Acinetobacter baumannii that we have in our collection on the Genome Portal? Here I have an example fragment of GYRA from a susceptible Acinetobacter baumannii. I'll go ahead and copy and paste this to get it ready for our next analysis. So what we'll do is take advantage of the portal's sequence search functionality located on the top banner of the Genome Portal right here. And then I'll go ahead and paste this sequence in. Finally, I can go to the bottom and hit the search button. On the left, you'll see that bases that match the genomic sequence of a genome published on the Genome Portal are highlighted in gray. So for example, this T right here does not appear in any of our reference quality genomes. When you roll over genomes on the right, which are the search hits, bases that match the genomic sequence of this genome in the search results are highlighted in this additional color. So you'll see here, bases that match this genome specifically in the Genome Portal are highlighted in yellow. And so we know that BAA 1710, BAA 1605, and 17978 are all susceptible genomes. But importantly, let's scroll down and take a look at 2803, BAA 2803. And here you'll notice a large fragment of the GYR gene that we pasted in does not match our global priority superbug, Acinetobacter baumannii. So you can see this substantial difference, and then we could go and download these individual genomes if we wanted to take a look at further differences between susceptible and resistant Acinetobacter baumannii genomes and perform further analyses on them. Finally, ATCC confirms the identity of our strains against highly curated database. To do this, we've partnered with OneCodex, an industry leader in bioinformatic bacterial identification, to verify our strain designations by using two approaches, both k-mer identification approaches and genomic distance approaches. Let's talk about the second one, the genomic distance approaches, for just a moment. So while developing this project, our goal was to sequence some of the most in-demand bacterial strains at ATCC. Many of these strains are very well characterized and fairly taxonomically stable. But we also know that our collection of over 18,000 bacterial strains, there are isolates that may have undergone significant taxonomic revision and reclassification, especially as we enter the genomics era. So we know that we wanted to specifically target a group where we saw that taxonomy was rapidly changing, to see how well we could identify strains that needed taxonomic updates and if we could appropriately place them into new taxonomic categories. So the genus Bacillus fits this bill pretty well, specifically the Bacillus cereus and Bacillus subtilis groups. In the past decade, 12 new species have been added to the Bacillus cereus group, and 3 new species have been added to the Bacillus subtilis group. What we did was sequence and assemble the genomes for 61 of ATCC's Bacillus cereus and Bacillus subtilis group strains, some of our oldest strains in the collection. We then took these genomes and measured genomic distance to the type strains of all taxonomically valid Bacillus. And what we found was incredible. 12 items are now being moved into Bacillus species described only in the last decade. 1 item is being moved into an older existing Bacillus species, and 9 items in the collection are being moved to taxonomically undescribed Bacillus species. Of course, we expect users would like to perform similar genomic distance comparisons, similar to the ones that we performed for Bacillus. And especially, we expect our users want to use the type strains we're making available on the portal that were part of our bacterial collection. For example, let's say you have an older isolate labeled as Streptococcus aeruginosus that has recently undergone next generation sequencing. Unfortunately for you, until the advent of sequencing technologies, Strep aeruginosus was difficult to phenotypically differentiate from other members of the so-called Strep aeruginosus species group, which also includes species like Strep intermedius. Let's go back to the Genome Portal for a moment and look at one of the search features available to users, specifically the ability to select just type strain genomes for use in analyses such as these. Back on the home page, you see the search bar where we can search for genomes. And when I click on this search bar, you'll see that I can enter a number of different search criteria. And we list multiple methods. You can search by species, search by genus, search by isolation source or catalog number, which we've already done. Another option you can search by is type strain. So I'll enter type_strain, colon, and then here we're going to indicate yes to select just the type strains from our database. Here our type strains are arranged in alphabetical order by taxonomic name. So let's just go to the last page to find Strep anginosus and Strep intermedius. Here we go, we found them. Of course, we could download these genomes right away from this page as such, clicking download and then download the assembly FASTA file for both of these. But let's explore a little of the S. anginosus genome. So I'll go ahead and click view to go to its genome page. So as I've shown you so far, we've performed a lot of quality control on these genomes, and we want to show some of this quality control data to users. We also want to give users assurance that this genome is free of contamination and is appropriately named. And we do all of that on the quality control tab right here. Here you see the sequencing quality metrics that this genome needed in order to pass our quality control criteria, along with the assembly quality control metrics. Of course, being free of contamination is of the utmost importance, but especially so for type strains which are used for taxonomic identification. Here you'll see genomic completeness and genome contamination metrics calculated from check-in. And you can see that for our S. anginosus genome, we have the entire genome present here as indicated by 100% completeness and free of contamination by an indication of 0% contamination right here. If you're interested in learning more about these metrics and how we apply them to our reference grade genomes, you can click on our approach to whole genome sequencing link at the bottom of the ATCC Genome page to read more right here. Okay, we have these genomes downloaded. Let's go ahead and compare them. Using these type strain genomes, we can calculate a metric called ANI, or average nucleotide identity, which is a measure of how closely two bacterial genomes resemble one another. In current bacterial taxonomic literature, two strains with 95% or greater ANI are considered members of the same species. So after measuring ANI between these genomes, we can confirm that Strep anginosus and Strep intermedius are indeed two different species, and we can see that our unknown strain is actually a strain of Strep intermedius, not Strep anginosus like it was originally identified as. To wrap up, today I've shown you a brief look at genomes.atcc.org, ATCC's new genome portal, which hosts reference quality genomes for our most in-demand bacterial strains. These genomes not only serve to authenticate ATCC's biological material with 21st century techniques, they are also a new tool for use by the research community, where we know this data will be used to make the next great scientific discoveries. We are adding new genomes to the portal constantly, so check back often to see what we've made available. If you're interested in a particular ATCC product's availability on the genome portal, or have any other questions about the portal that we don't get to today, please reach out to us. Thank you. I'll now take any questions.
Well, thank you, Andrew. In just a few moments, we will begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. The recorded webinar presentation will be available on demand on the ATCC website at www.atcc.org. Okay, and for our first question, how do I know if an ATCC product has genome sequencing available?
Okay, great. Thanks, Brian.
So first, I would say the easiest way to see if a genome sequence available is to go on genomes.atcc.org, go ahead and search for that particular product that you might be interested in. As always, you can go to atcc.org's main website, and you can also enter your product into the search field there. And as I've shown you guys already, you can see the genome sequencing available icon on all of our products that have genome sequencing available. There's another feature that I'd like to point out. If you purchased your product after September 2019, you can now view your products that have genomic data available by logging into your atcc.org account and visiting the My Genomes tab in your profile. If you purchased a product with genomic data before September 2019, these products won't be available on that tab yet. But like I said, as always, you can search on genomes.atcc.org.
Okay. Now, our next couple of questions are related to products and the Genome Portal. Now, why is it that some products have genome sequencing data while others do not?
Yeah. So as part of the Enhanced Authentication Initiative, our long-term goal is to offer genome sequencing data for as much of the material in our collection as possible. But as you might expect, whole genome sequencing requires a ton of effort and takes a lot of time. So we're moving through our collection as fast as possible while maintaining all of our high quality control standards. So of course, we make this data available to everyone through the Genome Portal as soon as possible, and we'll be releasing additional sequencing data and additional genomes as they come out and as they're made available. But we're making them available on a continual basis.
Okay, and so the other product, the other question, sorry, that's related to that. Why does a product not appear in search results when I search for it on the ATCC Genome Portal?
Yeah, I'd say there's either two reasons behind that. First, we haven't sequenced it yet, and in which, again, it wouldn't appear with the genome sequencing available icon on our main website. Alternatively, you could be searching for a derivative product instead of the main source organism, and on genomes.atcc.org, we group everything by the ATCC source organism. So for example, let's say you ordered a new nucleic acid product from us. You ordered 12228D5 and you enter 12228D5 on the Genome Portal, and you probably won't see anything appear. So what you'll need to do is just remember to yourself, the D5 product is the derivative product. So if you enter 12228 into the Genome Portal without that D5 suffix, then you'll arrive at the genome that you're looking for.
Okay, here's a question. Let's see. Are FAQs, frequently asked questions, available online around this?
Yeah, definitely. So on genomes.atcc.org, anytime you have a question about a particular process, you can scroll to the bottom. We actually have a lot of documentation on how we go about whole genome sequencing and assembly. So you can click on one of those links below. You can click on a link to our FAQs to help guide you through some of those points. And then if you have a question that's not answered by any of the links at the bottom of the Genome Portal, you can always click the Contact Us link as well, and that'll put you directly in touch with ATCC Technical Services.
Great, great. Okay, our next question is around access to the data in the portal.
Yeah.
Do I have to purchase a product to see the genome sequencing data for that product?
So definitely not. So as part of ATCC's pledge to enhance our authentication in science and biology, our whole genome sequencing is publicly available to the scientific community for non-commercial research purposes. If you're interested in using our data for commercial purposes, something like a licensing agreement, we'd be happy to hear from you. Again, just use that link at the bottom of the Genome Portal, get in touch with ATCC Technical Services, and they'll put you directly in contact with us, and we'll work with you from there.
Okay, great. All right, here's an interesting question. What criteria must the data meet before we consider the genome to be of reference quality and release the whole genome sequence on the ATCC Genome Portal?
Yeah, that's a great question. So as I talked a little bit about during the webinar, we go through an extensive quality control process. And really, like I said in the presentation, we differentiate ourselves by the fact that ATCC has this authentic, traceable material. And so obviously we go through all of our existing authentication processes here at ATCC when we start with the genome. Then we go through the extraction and sequencing and assembly process, and we use this optimized methodology that I talked about during the presentation. A little bit more about these exact quality control metrics. Like I said in the presentation, you can go to the quality control tab on any of our genomes, so you can see the particular metrics that are passing in order for it to be posted onto the genome portal itself. These are things like genome completeness needing to be 95% or greater, coverage being needed to be 100x or greater on a given genome, et cetera, et cetera. And if you're interested in the exact criteria that we use, we make that available. Again, go to the bottom of genomes.atcc.org, click on the link that says Our Approach to Whole Genome Sequencing, and that's a whole technical document that outlines all of our processes, so you can check that out for yourself as well.
Okay, great. So here's a couple of questions around crowdsourcing. Can my sequencing data be added to the ATCC Genome Portal?
So right now, no. As part of the Enhanced Authentication Initiative, we've made our pledge to authenticate our own diverse collection using the most current sequencing technology available. So kind of as such, our genome portal only includes genomic data that we here at ATCC have produced and importantly verified before it gets published to the portal.
Okay. Now, am I allowed to upload the genomic data to a publicly available database?
No. So the data use agreement specifically for the ATCC Genome Portal states that users are not allowed to update ATCC's sequencing data to publicly available databases.
Okay, and I think this one might be kind of related to it. Sorry if I missed it. If we have our own whole genome sequence, can ATCC Genome Tools help in assembly and analysis? Or does it have to be a sequence already in the database, in ATCC's database?
Yeah, so I'd say the central reason for the ATCC Genome Portal to exist is to act as a repository for all of the genomes that we've put together. If you're interested in some of the applying some of the same techniques that we use, feel free to go into our technical documentation, take a look at our best practices and apply that. But right now we're not performing genome assembly as a service to customers. We do it for our own products, and then we make our own products available through the portal.
All right. Now, here's a question about the pipeline, the bioinformatics pipeline. What tools do you use to read trimming and other sequence data QC steps?
So right now we're not divulging all of the details behind the exact tooling that we use in our bioinformatics pipelines. All the information that we are making available, again, it's listed in our pretty extensive technical documentation. So again, if you're interested in using some of our best practices, we make all that available, all that information available through our technical documentation. But we do use commonly available tools that people can use with a mix of our proprietary pipeline items.
Okay, cool. So basically what we can say is published on our website.
Yes.
Then. All right.
Exactly.
Okay, great. Okay, the next couple of questions are around functionality. One is, can I download the raw sequencing data as a FASTQ file? And the other is opening GBK files I download from the portal.
Sure, those are both really good questions. So right now we're not making raw sequencing data available as a FASTQ format. So we are providing the whole genome assemblies in FASTA format. In addition, we're providing the whole genome annotations in GBK format. So if you're not already familiar with GBK format, it's a plain text format that you can open, and it has information about the genome annotations. And typically it's used as kind of an input into your own bioinformatics pipelines. But if you're interested in opening these files, all you need to do is, if you're a Windows user, you'll need to decompress it using a commonly available decompression tool. If you're on Macintosh, you can just right-click it and select extract that particular file, and then you can just open it up with the text editor application. And of course, the GBK file format is compatible with any number of bioinformatics software that you might want to use.
Okay. Here's a sort of a future direction question. Is ATCC planning to deposit genomic sequencing data to publicly available databases?
Yeah, so right now the only place that you're going to be able to find our whole genome sequencing is through the ATCC Genome Portal. We're keeping it there because we want to keep it as closely tied to the source product as possible. Sometimes things can be quite confusing when you go onto publicly available databases, so we keep them tied to our products through genomes.atcc.org.
Okay. So now I actually have a question. Now you've basically created this great tool. You've kind of completed this really big project. Mm-hmm. What's next?
Yeah, that's a great question. So what's next is a lot of different things. We've just begun getting through the ATCC bacterial collection. We have over 18,000 bacterial strains. We're working our way through that collection right now, going through some of the most in-demand bacterial strains possible. So that's kind of our work expanding the bacterial collection. Things on the horizon. So right now we're deep into R&D on making viral genomes available for customers and making fungal genomes available. So those are on the horizon soon. But the ATCC Genome Portal isn't just restricted to that as well. Other things that we're looking into right now include things like ATCC's cell line collection and our cancer cell line collection as well. So this is not just an initiative limited to bacterial strains. This is something that we're working on expanding out and providing for all of ATCC's collection.
All right, great. Well, at this time, we will conclude our Q&A session. I'd really like to thank Mr. Frank for the excellent presentation. And thank you everyone for attending the webinar. Thank you again and have a great day.